Alright, this is part 2 from our membranes chapter, and in this section we're going to talk a little bit about transport across cell membranes. So we're going to start with the simplest, and then we're going to work our way towards some of the more difficult concepts. So the first one we're going to talk about is diffusion. So diffusion is really simple. It is just the movement of molecules from a higher concentration to a lower concentration. And it's really important, really important that you understand that this does not require energy because molecules are always moving. So as molecules bounce around and they hit each other, the collisions are going to cause them to go off in random directions, and what's going to end up happening is if there's more molecules in one area, they're going to be more likely to hit each other and get bounced around, and eventually what's going to happen is the concentration of molecules is going to diffuse. You're going to end up with similar concentrations all over the place on both sides of the membrane, not because they're going in one direction on purpose, but just because of random motion. And osmosis is just a special name for the diffusion of water, because water is so important. When we're talking about diffusion, we're usually talking about solutes, ions, covalent molecules, etc. When we talk about osmosis, now we're talking about a solvent, because a lot of the time the solutes are stuck where they are, but the solvent, water, can freely flow across the membrane, because if you recall, we have those aquaporins that allow water to freely flow through the membrane. Okay, so this is sort of showing a diagram. At the top we have molecules of dye, and what they're showing is that the dye is bouncing around randomly, but every time it hits a space, a pore in the membrane, it passes through. It's not that the dye can't go the other way. If we look at the second slide here, you see that some of the dye molecules are going the other way, but the chances of one hitting from the left are much higher than the chances of one hitting from the right. Eventually, once the dye molecules have spread out, the chances of one hitting from one side are equal to the chances of hitting from the other side. And so what we have is what you really should call dynamic equilibrium. It means that the concentration is the same on both sides. It's not the exact same molecules on both sides, because I gave the example in the classroom that we have two doors. If, let's just say, everybody was sort of bouncing around inside the room and there was also people outside the room, and every time somebody from outside got knocked in a door, somebody that was inside the room got knocked out of a door, we would maintain the same number of people in a room, because every time somebody went in, somebody else would go out. However, it wouldn't be the same exact people. So it would be called dynamic equilibrium, the fact that the molecules are still moving, but the actual concentrations aren't changing. So that's diffusion. Now, tonicity is a really important concept that we're going to talk about, and I mentioned this a moment ago, the fact that a lot of times the solute molecules cannot pass through the membrane. It's the water that can pass. And so this plays a huge role in equilibrium in a cell. So here's sort of your keywords. Hyper means more, hypo means less, iso means equal, and tonic is going to refer to the concentration of solutes. So the more concentrated one side is to another, that's going to be the hypertonic side. The side with a lower concentration of solutes is going to be the hypotonic side. And you can't have one without the other. I couldn't just give you a picture and say, hey, is this a hypertonic solution or a hypotonic solution? That would be like saying, is she smarter? You have to have a comparison. Is she smarter than who? And so the same thing here. You'd have to have two sides to compare to one another before you could say that one side was hyper or hypo. Now the rule, and you can just memorize this, is that water's always going to go towards the hypertonic side. So if you can figure out which side is hypertonic, that's going to be the way the water's going to go. And that's because if there's more solute, then proportionally there has to be less water. I'll show this to you in a diagram in a second. So let's start with the simplest one, isotonic. Isotonic is a situation where the concentration of solute on both sides is equal. It doesn't have to be the same solutes. If the solute is unable to pass across the membrane, then 20% of one kind of solute on one side is going to be the same as 20% of some other kind of solute on the other side. Because if 20% is solute, the other 80% is water. So that means both sides have an equal concentration of solute in comparison to water. Okay, so here's an example of what I'm referring to. Let's just pretend like right here is some kind of a semi-permeable membrane separating two sides. So on the left side of this semi-permeable membrane, I have a 20% sugar solution, and on the right side, I have a 20% salt solution. So that means that there's going to be these little sugar molecules bouncing around, and chances are that if one of these little sugar molecules hits this membrane, if the membrane was permeable to sugar, then the sugar would go through. And same idea that if on the right side of this membrane we have 20% salt, so here's our little salt molecules, and if a salt molecule happened to hit this membrane and the membrane was permeable to salt, then the salt would go through. But let's pretend like the salt and the sugar, that the membrane's impermeable to both of these. So anytime salt or water hit, or salt or sugar hit this semi-permeable membrane, they can't cross. But what we do know is, let's just say that this membrane is freely permeable to water. The reason I say this is an isotonic solution is because if water can pass, a solution's made of two things, solute and solvent. So if this left side is 20% sugar, the other 80% is water. And the same is true over here. If this side is 20% salt, the other 80% is water. Therefore, if the solutes are stuck where they are, the amount of water, the concentration of water on each side of this membrane is the same. And it doesn't mean everything stops, it just means that the speed at which water is going in one direction is going to be equal to the speed at which water is going in the other direction, and so we have what's called dynamic equilibrium. And that would be the situation in an isotonic solution. Okay, so if the solutions are not isotonic to each other, then that means that one side is going to be hypotonic, meaning it's going to be the side that has less solute, and the other side is going to be hypertonic, which is going to be the side that has more solute. Remember that if there's more solute, I just showed you this, that means there's less water, and therefore water's always going to go from higher concentration to lower concentration, so you can kind of just memorize that water's always going to go to the hypertonic side. So let me show you an example of this. Okay, so at the top of this I've got 3 beakers, and each beaker has we're going to pretend like this is a cell. And the cells, each of the cells has 10% salt solution. And then outside the cells we have different concentrations of salt in the solution. So in our first solution we can see that it's 10% salt inside and 10% salt outside. And assuming salt can't pass, we have 90% water on each side, so this would be isotonic. In our second situation, we see that we have 10% salt inside, but we only have 2% salt outside. So that basically means that outside we have 98% water, and inside of our cell we have 90% water. Now, remember that water is going to go from where there's more water to where there's less water. And so you can actually look at this and say, okay, that means water is going to go into the cell because there's more water outside than in. However, as I mentioned to you before, if you just look at the solutes, determine which side's hypertonic. In this case, inside has more solute, so the inside is the hypertonic side, and the outside is the hypotonic side. Water's always going to go towards the hypertonic side, because by definition that means it has more solute and therefore has less water. If you look at our third problem, we have the opposite situation. Inside the cell 10%, that means that that it's 90% water. Outside the cell 30%, that means it's 70% water. Again, if we just look at tonicity, the outside is hypertonic. The inside, this time, is hypotonic. And so water, in this case, the net movement would be out of the cell. The second problem here is what you see a lot of times. This is called a U-tube problem. So what they do is they have this U-tube with a semi-permeable membrane. They always have to tell you what can pass and what can't pass, and then they're going to ask you questions about this. So the first question here is to describe A versus B at the beginning. To do that, all you've got to do is just look at A and B and total up the solutes on each side. So if we add up the solutes on the left and we add up the solutes on the right, we're going to see that each side has a solute concentration of 3 molar. And so therefore, the answer to this first question is that at the beginning of the experiment, it's isotonic. Now here's a second kind of question they can ask you. Describe side A to side B with respect to glucose. So this time they specifically want you to just look at the glucose and ignore everything else. So if we do that, you're going to see that side B has more glucose than side A. Therefore, side B is hypertonic with respect to glucose. The second thing says, or the third one, if sucrose can't pass but glucose can, which way will it go? Okay, that's our standard definition of diffusion. Anything that can pass is going to go from where there's more of it to where there's less of it. So glucose is going to go towards side A because that's where there's less glucose. And now the hardest question, which way will water go? The way to answer this question is look at the solutes first. They're going to give you something that's pretty easy to figure out. It's not going to be something where you have to use math in order to figure this out. At the beginning of our experiment, it was isotonic, so that would mean that water would be going in both directions at the same rate. However, if we know that the glucose is going to go to the left, then that means that what's going to happen is that our left side, side A, is actually going to become hypertonic to side B. So if glucose goes towards this side, that's going to mean that the concentration is going to go up on this side. The concentration of solutes is going to go down on this side. And that basically means that water is going to go towards the hypertonic side, which is going to be side A. And what we would see at the end of this experiment would probably be that the water level would be higher on side A and lower on side B. So that's how you solve one of these YouTube problems. Alright, so now that we've discussed the fact that water can freely flow into and out of cells, because remember they've got these aquaporins that let water through, we know that what's going to happen is if it's not an isotonic situation on both sides, water is going to either be entering a cell or leaving a cell, and that obviously is going to cause a problem because cells need homeostasis. So osmoregulation is basically the term for maintaining water balance or controlling the water balance in an organism. So let's start with freshwater. In freshwater, you have organisms that are going to be hypertonic to their environment. In other words, they're going to have more solute in their cells than freshwater is going to have on the outside. So the paramecium is an example we talked about in the last chapter. Paramecium has a special vacuole called the contractile vacuole, and it basically just squirts out excess water. Freshwater fish make lots of urine. That's their way of getting rid of it. It actually works to their advantage because one of the waste products they make is ammonia, and ammonia is highly toxic. And when we make ammonia, we have to expend energy to turn it into urea, which we then excrete in urine. Fish don't have to use energy to do that because since they have a constant water supply, they can just constantly flush the ammonia out of their bodies. So by making lots of urine and by they can basically survive and use it to their advantage. Saltwater fish have the opposite problem. They're basically living in an area where the area would be hypertonic to them. The way that they solve this problem is, number one, they maintain high solute concentrations within their body, because remember, the solutes don't have to be the same, just the water concentration. So they typically maintain high urea concentrations to match the high salt concentrations on the outside. Another thing that a lot of them do is they actually expend energy excreting salt. And we see this even in, like for example, saltwater crocodiles who have to drink salt water. They have salt glands to get rid of excess salt. Okay, in plant cells it's a little different. Plant cells have cell walls and central vacuoles. So what they're going to do is they're going to basically absorb as much water as they possibly can. It's going to create the pressure that they need on their cell wall that actually maintains turgor pressure, meaning it's what makes the plant leaves and stem and all of that stiff. Unlike animal cells that are normally in isotonic solutions, plants are typically, their cells are typically hypertonic to the external solution because fresh water is constantly coming in at the roots. And in fact, if a plant cell is put in an isotonic environment, it will probably get somewhat wilted, which the word for that is flaccid. Extreme condition would be if you were to put a plant in salt water or under severe dehydration. if enough water is lost, what will actually happen is the cell membrane will pull away from the cell wall, and that's called plasmolysis. And we saw that in the lab with the Elodia. It will kill the cell.